Alright, hey guys, this is part 6 in chapter 2, and this is the last part of what we're going to go over in this chapter, getting you prepared for that test next Monday morning or Monday afternoon. So we've been talking about macromolecules, and we've been talking about carbohydrates and fats and proteins, and the last of those is nucleic acids. So that's what this section is about. Hopefully it should be some information that's familiar for you because you did that project and you talked about this in your project. But we're going to do a brief overview of it so you can kind of remind yourself and get used to looking at this again. So first thing we're going to talk about is what nucleic acids are. Like I said, they're macromolecules. They have hydrogens, oxygens, and carbons, just like proteins, fats, and carbohydrates have. They also have nitrogens, just like proteins do, but the one difference is that they have phosphorus in them. So if you see a phosphate group in a molecule like this, then it's a good indication that that's a nucleic acid. So there's the things that you're looking for in the structure of the nucleic acid, like we talked about before. You should know the structure of all these different molecules. We talked about the structure of the carbohydrates and the structure of the lipids and the structure of the proteins. So here we're looking at the structure of the nucleic acids. It has a 5-carbon sugar right here, and that can be ribose or it can be deoxyribose, and those are sugars. So each corner here has a carbon bond there, and then it has a phosphate group, which you can see out here, and it has a nitrogenous base, which is off the side over here. So that's what creates that formation to make a nucleic acid. So the nitrogenous bases, there's different types of nitrogenous bases, and you may recognize these from previous science classes. You're looking at purines and pyrimidines. Purines are adenine and guanine, and those are found in DNA and RNA, right? You see those in DNA and RNA. Pyrimidines, these are smaller single ring structures. Cytosine, thymine, and uracil, those are considered pyrimidines. So cytosine, you're going to find that in DNA and RNA also. Thymine, you're only going to see that in DNA. Uracil, you're only going to see it in RNA. So there's the nitrogenous base options when you're looking at structures of the different nucleic acids. What do they do? Well, definitely storing genetic information, because we all know that DNA is where our genes come from, and that's where we determine what we're going to look like and how our bodies are going to form and what different tissues are going to be created. We talked a little bit about this when we talked about proteins and talked about protein synthesis a little bit, but so they store that genetic information. They also transmit genetic information, and we use RNA and DNA to make that happen. RNA has the sugar ribose, and DNA has the sugar deoxyribose, and that's where they get their names from. So ribonucleic acid is because of the ribose sugar, and deoxyribonucleic acid is because of the deoxyribose sugar. And that's that 5-carbon sugar that I was talking about in the structure earlier. DNA is the genetic material found within the nucleus. So when you look at the cell structure, you should remember this from other science classes. You have that nucleus in the middle. The main function of the nucleus in the middle is to store that DNA. And that DNA can replicate itself. It can create exact replications of itself within that nucleus, but the nucleus contains the DNA. It won't allow the DNA to leave that area. It holds it in there. DNA also provides instructions for building proteins, and we talked about that when we talked about proteins during the process of mitosis, transcription, translation. That creates proteins, which creates different structure and function in our body. So nitrogenous bases. Talking about this again a little bit. In DNA, the 4 nitrogenous bases that you have are adenine, guanine, cytosine, and thymine. those 4 options are available in DNA. They pair with each other to create that double helix of the DNA, which I'm sure you've heard that term before, double helix. It's two-sided, and when you have the 2 strands that come together, they connect at those nitrogenous bases. So you have adenine and thymine, those pair together. Guanine and cytosine, those pair together. And that's always the way it is. G with C and A with T, or T with A and C with G. It's always that way. And that's in DNA, only in DNA. So structure of DNA, you have that phosphate group, and then the deoxyribose sugar, and the nitrogenous bases, which are the A, T, C, and G, we see in the middle. So here's the sugar phosphate backbone. There's the nitrogenous bases in the middle. Alright, so here's a little better picture of it. Adenine nucleotide. Here's the phosphate group. Here's the deoxyribose sugar, and here's adenine, the nitrogenous base off the side. So this right here is one side of this double helix. So for example, right here, this guy, here's the phosphate group and the deoxyribose sugar over here, and then that's the adenine, this thing right here, coming off there. This one over on the other side is going to have a ribose sugar, a phosphate group, and then a thymine nitrogenous base connected to it. And because of the attraction between those 2 nitrogenous bases, that's what holds that backbone together. And like I was saying already, DNA, it looks like a twisted ladder or like a double helix. It twists around itself, and all those nitrogenous bases are binding those 2 strands together, which creates that double helix. So here's the double helix again, another example, and then this is what it looks like. And it actually does, I mean, of course it's not as pretty as this, but it does actually look like this under a microscope if you're looking at DNA in the nucleus. So in DNA, which bases pair together? We already talked about this a little bit. Adenine with thymine, spells at, and then guanine always pairs with cytosine. G and C look very similar to each other. G and C, those look like they go together. Those 2 pair together. So this is always true. It never happens any other way. A is always with T, G is always with C. That's it. And that, again, this is only in DNA though, because in RNA there's a little bit of a different sequence there, a little bit of different combination. So you got complementary bases, A and T, G and C. They're considered complementary bases. They're held together by that hydrogen bonds. And we talked about those in the early parts of this chapter. We talked about hydrogen bonds and how that creates a 3D structure. And there's kind of an uneven combination there and sharing there, and water can be used to break up those bonds. So we talked about that a little bit already, but that's what's holding them together, those hydrogen bonds. You can see how they're kind of weak bonds too, and they have to be, because when we do get to the point where we have to create protein, then we need that DNA to split in half. Alright, so what is the complementary DNA strand if the original DNA strand is AGTCTA? Now you're going to have to, that's one side. This is one side of the helix. You're talking about what's the other side of the helix, and that's what you're trying to match up here. So A always matches with T, right? So you're going to have a T here. G always matches with C, so you're going to have a C on the other side. T is always with A, so you're going to have an A over there. C is always with G, so a G over there. T with A with T. So that's what we got, T, C, A, G, A, T. And that is a complementary strand, which will stick right up over here. RNA is located outside of the nucleus. So the DNA is inside the nucleus. The RNA is outside, but it starts inside the nucleus. And the way that happens is the DNA has to be split, the double strand has to be split, and it recreates a part of itself to create RNA so that it can go out into the cytoplasm and create proteins. It does that at the ribosomes. So hopefully some of this is jogging your memory from when you took other science classes like biology and stuff, because you should have learned about this already. But that's the basic process. So RNA has ribose in it, and it's a single-stranded, and it contains uracil instead of thymine. So that's what I was talking about with the nitrogenous bases before. And RNA is different. You still have guanine, you still have cytosine, you still have adenine, but you no longer have thymine, you have uracil instead. So RNA contains these here, no thymine. So this is RNA over here, and this is DNA on the right. And you still have the nitrogenous bases, but they're different here. Except the first three are the same, but you have uracil instead of thymine. You still have the sugar phosphate backbone, which in this case in RNA is ribose, and you also still have the phosphate group there. So three main types of RNA. You have mRNA, which is the M stands for messenger, and the reason why it's called that is because after that transcription process, you have a new strand of RNA like this that is just like half of the DNA, and that's going to go off and go out of the cytoplasm to be able to create proteins. So it's called a messenger RNA because it's taking the message from the DNA out of the cytoplasm to create proteins. Then you have ribosomal RNA, or rRNA. This is a major part of the ribosomes, inside the ribosomes, and that's where ribosomes is where protein synthesis occurs. So again, hopefully you remember some of that from biology. And then the last one is called transfer RNA, or tRNA. And what this one does is it actually brings amino acids over to the ribosomes so that the mRNA and the ribosomes and the tRNA can all come together and create a strand of amino acids. And like we learned in the last section, what's a strand of amino acids? It's a protein. So you need those amino acids from the tRNA to produce it. And we talked a little bit about this in the protein section too. These right here, this is called an anticodon. On the rRNA, you're going to have a codon, which will match this or be a complement to that, and that's how the tRNA knows which amino acid to bring. And that's that's signaling that's going on. So again, this is a really intelligent design that's happening inside our body all the time. And ATP is the last nucleic acid, adenosine triphosphate. It's called a triphosphate because it has 3 phosphate groups coming off of it like this. It has an adenine over here, that's where the A comes from, and then the ribose in the middle, that's the sugar that holds it together. ATP, again, is our energy, and this is considered a nucleic acid, and glucose is what we use to create that ATP. And we talked about that before, and that happens in the mitochondria. Glucose is very important for that to happen. And where do we get glucose? Carbohydrates. So very important. So the structure, we have adenine, ribose, and a triphosphate over here. So that's where that name comes from, adenine triphosphate, adenosine triphosphate. And I told you before, that's potential energy. When it's split and that phosphate group comes off, that becomes ADP, or adenosine diphosphate, and that's where energy happens. And that's what we use to create energy for different systems and functions in our body, so that we can use it after we've broken it down in food and fuel. So that goes through all the macromolecules, and we are now done with chapter 2, ready for the test on Monday.